Hello everyone, I am Chen Baoying with matrix number 74423. Now I would like to present about the steps of virus replication. Viruses, also known as the infectious particles. What is virus replication? Virus replication means that the virus infects the target host cells by replicating themselves. There are 6 steps included in the virus replication, which are attachment, penetration, transfection, replication and synthesis, assembly, and the last step is release. The first step in virus replication is attachment. Attachment, also known as adsorption. Attachment means that the virus attaches on the outer membrane of host to infect it. In addition, the antigens on the surface of virus need to fit the receptor site of host. The fit is species-specific, where only a type of animal cells and plant cells are infected by a particular virus. The second step in virus replication is penetration. Penetration means that the virus injects its genome into the host. Most viruses are drawn into the host by endocytosis upon attachment. There are some types of viruses can move directly through the pores of the host membrane. However, there are also some that remains outside of the host membrane but injects its DNA into the host. This is all from me. Thank you.
Hello everyone, my name is Chai Chia with matrix number 74358. Now I'm going to present the transfection. The transfection, which is the third step of virus replication. During this step, the viral DNA is inserted into a specific site on the recipient cell by integrase. The enzyme acts this by cutting the circular plasmid DNA, then splicing in the new DNA and repairing the two sites. The asexual transfer of DNA enables the occurrence of DNA recombination. Therefore, it provides new genes to the new host. At this point, they are favored by low pH values. The next step is replication and synthesis. The first step in gene expression is mRNA synthesis. DNA virus replicates in the host cell's nucleus, except post-virus replicates in the cytoplasm. On the other hand, RNA virus replicates in the cytoplasm except influenza virus and retrovirus. During this step, RNA polymerase synthesis mRNA. Once mRNA is synthesized, it will be translated into viral proteins by host ribosomes. The polymerase will synthesize lots of copies of viral genetic material for the progeny virus particles.
Assalam and hi, my name is Leung Wei Ning and my matrix number is 75406. The fifth step in the virus replication is assembly. After all the necessary viral components are synthesized, the virus start to assemble themselves. The viral DNA is firstly packaged into the head for the bacteriophages. Next, it is followed by the protein scaffolding. The protein scaffolding is constructed to assemble the tails of the bacteriophages. Then it is followed by the attachment of the tails of bacteriophages. The protein building blocks, which are capsomes, are added to enclose the virus. The last step of the virus replication is release. The progenies are extruded and budded off from the host cell after they successfully assemble themselves. The host cell will eventually undergo lysis of the plasma membrane. The process is repeated for several times when the virus is released and transmitted to other parts of the body of the living organisms. The virus will infect other host cells but undergo the same steps, which is the virus replication steps. Thank you. Hope you enjoy our presentation.